Okay guys, so tonight what we're going to be talking about are tools that we'll be using a lot in a microbiology lab. So things that you guys will be using a lot in a nursing environment, in a research lab, in a hospital, things like that. So this is an introduction into how these things work, kind of some of the things that are fairly common in labs, things that you guys will see a lot. So let's go ahead and get started on it. So one of the most common things for a microbiologist to encounter and kind of one of the most introductory basic techniques in a microbiology lab is something called a Gram stain. So Gram stains were discovered in the 1880s by a guy named Hans Christian Gram. He's a Danish physician, and he was looking for a technique that he could use to stain different types of tissue that would allow bacteria to stand out against, say, like lung tissue or like liver tissue or something like that. So we could take a sample of someone that was had a lung issue or something and stain it with this particular stain, and the lung tissue would be unstained or maybe stained purple, and bacteria would stain green or pink or something and stand out very obviously from that particular tissue. So he kind of stumbled upon this method kind of by accident on how to stain bacteria. He was looking kind of more to stain different types of tissue and different types of lungs tissue and things like that, not so much for bacteria. I mean, he just kind of accidentally stumbled upon this technique that allowed bacteria to stand out from tissue. So kind of once this technique was kind of started, where people had realized that you could use different types of stains and different types of liquids and things like that to make bacteria take up colors, and stand out from tissues and things like that underneath a microscope. They refined these techniques, added to them, and took away different things that may or may not have worked, and refined them a little bit. And eventually they came up with a Gram stain. So we'll get into what a Gram stain is later. I like to start these lectures off with a little bit of history, kind of what people were thinking at the time, kind of why these techniques came around and stuff like that. So anyway, so these are the 6 or 7 I's of microbiology. We'll talk about these a little bit more towards the end of this lecture. This is kind of the steps of what to do in a microbiology lab, kind of starting from getting a sample from your patient or from the environment, going through the growing that, isolating it away from all the environmental samples that may be out there, the extra bacteria in the environment or the ones from your patient, growing it, identifying it, and things like that. So we'll talk about these a little later as we get towards the end of this lecture. But first things first, we're going to jump into one of the most important tools of a microbiology lab, and that's a microscope. So I'm going to bring the mouse on the screen here so we can talk about some of the pieces of this. So microscopes are really easy. Every single college or university or high school, things like that, has one of these types of microscopes. This is called a compound microscope. There are lots of different types of microscopes out there. I've got a slide on that in just a second. This is one of the most common types of microscopes that are found in those research environments, schools, and things like that that I just mentioned. So compound microscopes get their name because the magnification compounds on each other. So the eyepieces up here have a magnifying power of 10, as you guys can see there. The objective lenses down here have a magnifying power as well. So this magnifying power times this magnifying power compounds upon itself to give us, say, this is 10, one of these is 4, so 4 times 10 would be 40. So it compounds on itself to give you 40 times more magnification than what you could see with just your naked eye. So let's go through the parts of these real quick, what they do. Pretty much every single compound microscope out there on the planet looks the same. They all kind of function the same. It's kind of like cars. Things may be in a little different position, or they may move a little different, but they're all basically the same. So like the on and off switch, or maybe the light adjustment things may move around from place to place, or maybe on the side or something, but the microscopes all basically are the same. So anyway, top down, eyepieces. This is what you look for. They're also called the ocular lenses, the eye lenses, things like that. You put your eyes up here. These things are adjustable. They can move back and forth, up and down, to allow you to get a good fit on your eyes. They are removable. They have a magnification power of 10. So that's where base magnification. So you can pull those things out and clean them if they get dirty from time to time. So down, we have the body tube. And this is going to be the thing that connects the whole microscope together. It connects the eyepieces. It connects the rest of the microscope to the important parts. On down, we have the nose piece. This is the round portion right here of the microscope. This is going to be the revolving portion that holds the objective lenses in place. So this thing's going to spin around the nose piece. So the objective lenses, what these things are, there's usually 4 of them. Sometimes they have more, sometimes they have less, but most of them have 4. Our microscopes at Parsons have 4. Most of them do as well at most research schools. So anyway, there's 4 objective lenses here. These things are actually going to be doing the magnification. So you got 10 up here. These usually are in magnification powers of 4, 10, 40, and 100. So 40 times bigger, 100 times bigger, 400 times bigger, and then 1,000 times larger than what you could see with your naked eye with the compounding effect. So bacteria can only be seen, for the most part, under the 1000x objective lens. You can kind of see them underneath the 400x, not particularly well. You can kind of make them out, but you really need to go underneath the 1000x to see them well. This is also called the oil objective lens. I'll talk about what that's for later. The 4x is called the scanning objective. The 10x is usually called the low power. The 40x is called the high power. So anyway, the stage down here, this is what actually holds the slide that you're looking at. You can move this thing back and forth. There's a little lever on the other side of the microscope over here that allows you to do that. You see the stage clips right here as well. This is the thing that's actually going to be holding the slide in place. Underneath, you'll see this little lever. It's called a diaphragm. Excuse me. You move this thing back and forth. You see there's this little track right here. They slide back and forth along this little track. And it kind of works like the shutter on a camera. The more open the diaphragm is, the more light that goes through the microscope. The more closed it is, the less light that goes through. And you can open that and close it to adjust the darkness and sometimes bring out the more better focus on your specimen. And here we have the condenser. It sits underneath the diaphragm. You can kind of see this little silver screw here. You can pull the condensers on and off, change them out. And the condenser works kind of like a magnifying glass for little kids that burn ants in their front yard. It condenses the light beam from the light source down here. It's like a light bulb. It condenses the light beam into a very, very, very fine, from a big, large V shape, just a big, large straight line that would come up from the light. It condenses it into a really, really, really fine little beam that's sent right through the specimen, up into the objective lens, and then out the eyepieces right there. So on the rest of the way over, we have the on and off switch. They're usually in the back, and then the power cord's pretty easy. So you have these 2 knobs over here. You have a big one, and inside of the big one, you have a smaller one. So the big one's called the course adjustment knob. It makes really, really, really large adjustments in the position of the stage. If you grab the course adjustment knob and you give it a big spin, this stage is going to move up and down very quickly. And that's how you adjust the focus in the microscope. You have to be very careful with the course adjustment knob. If you move it too quickly, or it's too close to the slide, it can drive the objective lens through the glass slide and it'll send glass shards everywhere. Not so much fun. Underneath that, or inside, I should say, of the course adjustment knob, we have the fine adjustment knob right here. The fine adjustment knob makes little tiny, very precise, very small adjustments in the height of the stage. And that allows you to bring in specimens into a very, very, very precise, very sharp focus instead of a kind of a blurry. You can refine them with the fine focus knob here. So anyway, we have the stage clip. I already talked about that. The arm, that pretty much covers the parts of our microscope. They usually have a wheel or some kind of lever over here, which are maybe on the other side, sometimes in the front, which will adjust the brightness of the actual light bulb itself, so you can dim it or make it brighter and things like that. So once again, every single microscope out there that's a compound microscope pretty much functions, looks, and works pretty much about the same. So what is magnification? So magnification is just the ability to make things seem bigger than they are. So I talked about the concept of compounding magnification using the eyepieces here and compounding it with the objective lenses. And that's referred to as the total magnification. So the objective lenses are 10x, the eyepieces are 10x, the objective lenses are 4, 10, 40, or 100, so you just times 10 by whatever. So it would be 40, 10, 40, 400, and then, or 100, 400, and then 1000. So another really important property of a microscope is the ability for it to resolve one thing from one another underneath the microscope itself. So the concept of resolution. So if you know anything at all about how TVs work, or computer screens, or cameras, things with pixels, you'll know that the more pixels that are found per square inch, the better the actual picture will come out. So microscopes work the same way. the better they can resolve, the better the pixels of the microscope is kind of the better the picture that you're going to get. Now, microscopes don't really use pictures per se. They don't use pixels. What they use to take pictures are light beams. So some microscopes use light, compound microscopes do. Other types of microscopes use electrons or different rays of light with smaller and bigger wavelengths and things like that. So resolution from a microscope refers to its ability to resolve one little tiny cellular structure from one another, or one cell from one another, things like that underneath the microscope. So how this works. So light beams are quite large. They're very wide. If you've ever seen the little zigzaggy waves of the light spectrum and things like that, you'll know light beams are quite large. So if you're trying to look at your hand or something like that underneath the microscope, if you're using light beams to take a picture of your hand, they can't fit down in between the little narrow cracks. So imagine these big balls as a light beam. They can't fit down in beside the little narrow cracks of your fingers. They can't get around in the detail. They just can't do it. So light doesn't penetrate as well as smaller electrons and things over here. So these are tiny little electrons. Electron scanning microscopes use electrons to take pictures and not light. So they're significantly smaller than light beams. So you guys can see here that the electrons have the ability to get down inside of all these little details and make super, super, super fine detailed pictures between, like I say, of your hand, when compared to like large light beams that just don't have the ability to get down in there. They cannot penetrate down into the details. They just can't do it. So resolution is the ability to do this. This has much better resolution than this. So even if you might be looking at them, as you guys can see, on the same magnification, there's no difference in the size here. They look different when you take the picture up here because of the ability to resolve. This has a much lower resolution power than this does. Microscopes work the same way. Whatever they use to quote-unquote take the picture with to allow you to see, if it's large, you're not going to get as good of a picture as you say compared to something that uses electrons or something smaller to take a picture. So this is the pixelized version of that over here. You guys can kind of get the same idea. So you can increase resolution on microscopes by just using different types of microscopes, using a different type of lens, using a different type of condenser, or opening and closing the diaphragm, filtering out different spectrums of light. Blue light tends to be the best for resolving power. It has a very small wavelength, so it can get in between places that larger wavelengths of light can't. So this is the concept of resolution here in actual practice underneath a microscope. So this is a light microscope. You guys can see here it uses those light beams, really large things to take pictures with. So it's 450 times magnified, 450 times magnified, pretty much the same picture. But over here, our electron microscope uses the electrons, significantly smaller things to take photographs with. So you'll see over here, very blurry. It can't get down in between to give good details, whereas over here, not blurry at all. Really good details. You can see kind of even inside of the little mitochondria and things inside of the cell, which is pretty cool. Whereas over here, you just can't. It's just kind of a blurry blob. You can't really tell what you're looking at. So sometimes you need microscopes that need to take really good pictures when you want to see things inside the cells, and sometimes you don't. You just need to see what they look like, what shape they are, so you can tell that's round. This is different types of microscopes have different resolution power, and whatever you need to know from your research or whatever you need to define from the hospital to figure out and things like that, you'll have the microscopes that are best used for that, probably available for you already. So one of the things that are used a lot, in microbiology labs are oil. So I will refer to the 100x, the oil lens that's eventually the one that's got the 1000 magnification, as the oil lens. And most people do as well, the oil immersion lens. Oil is used a lot in a microbiology lab to kind of help resolution, also to increase the light that comes into a microscope. So the closer you get to the actual specimen itself, the actual slide of the microscope, when you spin the microscope around, you'll notice that the objectives get closer and closer and closer to the microscope itself, to the slide, I should say. The closer they get to the slide, the less light that is able to go into the objective lens. The farther back you are, the more light that's available. The closer you get, the less light that there is available for you. So the farther you get away, the brighter it is. The closer you get, the darker it gets. It's kind of counterintuitive. So the closer you get, the darker it gets, which is the exact opposite of what you need. You need more light to be able to see things that are really small. So what we do is we add a little drop of oil that sits in between the lens itself and the slide. So you can see over here we don't have one without oil, and over here we do. So what the oil does is it bends the light beams. It kind of tricks the light beams into thinking, oh, we actually have a lot more weight. It bends them. And it forces them to go up inside of the objective lens instead of out into the environment over here without the oil. So the light source would come up, hit the slide, deflect off to the side. Whereas it comes up over here, hits the slide, deflects off, and then is directed by the oil up into the objective lens. So oil allows way more light to come into a microscope the closer you get, whereas when you don't have it. Now, oil is only supposed to be used on the oil immersion lens itself, the 100x lens on most microscopes. It can mess up the glass inside of the other three lenses. So the oil immersion lens, the 100x, is the only lens that's supposed to have oil added to it. So there's a couple of different types of microscopy that's found in microbiology labs. So microscopy is just using a microscope or using microscopes to do things, or the study of using a microscope and things like that. So one of the most common types of microscopy that you will encounter out in the research environments in schools and things like that is something called bright field microscopy. So bright field microscopy, so this is a bacteria, or these are yeast cells underneath a microscope. So you'll see here, these are the cells themselves, and this is the background. So bright field microscopy allows the specimens to stand out against a brightly lit background. So most of the time the specimens are slightly darker. They cast shadows, they block some light, so they appear darker than the unstained, unblocked environment behind them. So this allows you to see them. So these guys are clear, they don't have any color inside of them, so you add a little color or you add the light, and they'll block some light, cast a shadow on themselves, which allows you to see them. So most microscopes, most compound microscopes that you'll find in research labs and schools like that tend to work on bright field microscopy, especially for schools and things like that. So dark field microscopy, I forgot to add, this is used a lot for living specimens, large things like this yeast. It's probably alive. You want to look at it move and do its thing. You can look at amoeba and stuff underneath the microscope as well, watch them move around. And it's all used a lot for stained specimens. So in microbiology labs, we stain a lot of the specimens. We kill them and add a lot of color to them. They're so small you would not be able to see bacteria without doing this, adding some color to them. So this is the type of microscopy that's used a lot to see those stained specimens. So our next type of microscopy is called dark field microscopy. It's the exact opposite of bright field microscopy. So you add a dark background, you add a filter or a stain to the background, and it colors the background really dark. And it allows bacteria to stand out against that black background. So the background absorbs all of the light, and no light's able to penetrate through. The bacteria are quite clear, they're see-through kind of, so they can move the stain out of the way as they move through the background, and then the light will come through them. So you can see their shape coming through the background as they move. So the second this little guy moves up here, the black little background will fill in behind him, so he leaves kind of a little path behind him for about 2 or 3 seconds before the stain will cover it back up. So this is used a lot for bacteria that are living. You don't really want to observe a lot of living bacteria underneath a microscope. They're kind of dangerous to do that with, especially for beginners. But there are some that you absolutely cannot stain. They will just destroy themselves if you try to stain them. It just breaks them down. They just can't do it. So the only real way to look at them is alive. So this is the type of microscopy technique that's used to do that. Can't stain them. They have to be alive for you to look at them, or else if you kill them, they break apart, they dissolve. You can't add stain to them because they dissolve. So this is the only way that you can look at them. So this happens to be Lyme disease, Borrelia burgdorferi. It's a very common one that is available only from dark field microscopy. Phase contrast, it's not as common. uses different wavelengths of light, which is cool. It's kind of if you've ever seen ghost shows, you see them, they use the full spectrum camera that uses different wavelengths of light that humans can't see. This one does kind of the same concept. It passes different wavelengths of light through the specimen from underneath that allows different structures to be illuminated at different wavelengths and things like that. So we can look at different phases, different depths inside of the organism at the same time, which is kind of cool. So fluorescence microscopy is really neat. You can use this a lot to tag different types of infections. This is cool. So this could be lung tissue, or excuse me, this would be intestinal tissue, or maybe, yeah, it could be anything really. Could be a water sample or anything like that. You add a special dye to it. Any type of bacteria that you're looking for will be tagged with this fluorescent dye, and then all you have to do is shine a UV light on it underneath the microscope, and the bacteria will show like glow sticks, which is pretty cool. So this could be a water sample where they've tested it for E. coli. Down here, the same thing. And what they did is they just poured some fluorescent dye on the water sample, put it underneath the microscope, hit it with the UV light, and then poof, there's the bacteria right there. You can see you got E. coli in the water sample. Pretty neat. Works a lot of the same kind of way when you're looking for bacteria in a body tissue and things like that. You can do it for lung tissue and things like that. I'm not going to talk about this one too very much. Electron microscopy is super cool. This one uses electrons to take pictures, which is super neat. So it has really, really, really high resolving power, uses very, very, very tiny pixels to take photographs. So it can get really, really, really high magnification, really, really, really highly detailed pictures of the actual surface of bacteria. So you would never see bacteria this detailed up and close. So the bacteria cells right here. You would never see bacteria this close, this detailed, to be actually seeing the bumps of their surfaces, the little grooves and things that they have on them, with the types of microscopes that you'll find in most schools, compound microscopes and things like that. So electron microscopes are really, really, really good at taking really detailed pictures. So anyway, bacteria. When you want to look at bacteria underneath a microscope, it's really difficult to just take them off a petri dish, stick them on a slide, and look at them underneath a microscope. They're alive. Some of them produce toxins. You don't really want to use living bacteria underneath a microscope. You could contaminate yourself for the microscope, which could contaminate whoever uses it later. You could contaminate the environment. Anything like that could happen. So we don't really like to use living bacteria underneath a microscope. So most of the time, what we're going to do is we're going to stain them. We're going to add some kind of a dye to them, or pigment that will penetrate the inside of the cells and give them color so we can see what they look like. But not always. So what we're going to do is we're going to talk about the two different types of stains. This one tends to be alive. This one tends to not be alive. So wet mounts is probably almost always alive. Well, anyway, so a wet mount sounds exactly like what it is. If you want to look at something move, you want to look at something swim, eat, function, do its actual day-to-day thing, what you're going to do is you're going to want to take a little sample of water or a little sample of agar or whatever, put it on a slide, and then watch the little small microscopic organism swim around through that drop of water. Watch it move, watch it do its thing, watch how it navigates, watch how it responds to food and things like that. It gives you quite a lot of details about the size of this organism, how it moves, things like that, what its shape is, how it's put together, and things like that. And then you have fixed mounts. So fixed mounts are dead bacteria cells. So I mentioned we don't really like to look at living bacteria underneath a microscope, especially for newbies. So we're going to kill them. So what you're going to do is you're going to take a little sample of bacteria from your a petri dish and put it on a slide, and then you're going to kill them with using heat most of the time, or some sort of acid and things like that. And then you're going to add a stain to it to allow you to see the bacteria, which otherwise would just be clear against that glass slide. So before you can stain bacteria cells, so this is going to be a fixed mount. So I'm going to skip over the wet mounts. You guys got the idea on that one. Really easy. Just take a drop of water, put it to a slide, and watch the stuff inside of it. So fixed mounts are going to be what most bacteriologists and microbiologists will do most of the time in their careers. some variation on this. So every single fixed mount on the planet is going to start out with the exact same procedure, something called heat fixing. So as I mentioned, bacteria are alive when you pull them off of a slide, and you don't really want living bacteria on a slide, kind of dangerous. So what you're going to want to do is you're going to want to kill them. So that process is called heat fixing. So it does one of two things. One, if you guys have ever gotten anything slightly wet or slick or something on glass and you add water to it, it just washes away. So this is the same concept here. If you just stick bacteria on a slide and then you try to add some stain to them, it's going to wash it away. So when you go to look at it, the bacteria just washed away by the liquid stain. So you're going to want to make sure that you stick them to the slide. So this process does two things. It kills the bacteria, which is what we want. We don't want live bacteria. And it's also going to glue them to the slide. So what you do is you take a little sample of your bacteria, you smear it on a slide, or usually put it in a little drop of water and smear it around in the drop of water. And then you hold it over a Bunsen burner or an incinerator or a slide warmer or something like that, which will dry out the water. And the same time, what it's going to do is it's going to cook the bacteria as they get dried out. It's going to dehydrate them. It's going to heat them up. It's going to cook them, which kills them. And it's also kind of melt their surfaces and stick them to the glass. So when you add some liquid to it, some stain, they won't just wash away. They've kind of been glued to the surface. So every single type of fixed mount pretty much starts out the same way. So there's three different types of stains that are used a lot in microbiology labs. You have something called a positive type stain, a negative type stain, and a structural type stain. When you look at bacteria underneath a microscope, you can't see them by themselves. They're clear. You have to add some color to them to see them. So what you're going to do is you're going to add some stain after you've heat fixed them. You're going to heat fix them, melt them, kill them, stick them to the slide, and then you're going to add some color to them. To put a couple of drops of liquid over the top of that slide, let the liquid penetrate, and then you can wash the liquid off and dry the slide again and look at them underneath a microscope. So positive type stains. This is a positive type stain underneath the microscope. This is Staphylococcus bacteria underneath a microscope. So what you'll see is they have heat fixed the bacteria to the slide, and they've added stain. So what they did is they let it sit for a little while, and the stain itself penetrated the bacterial cell walls. So the bacteria come out as nice little purple circles. You can see just the shape of the bacteria, whereas the slide, the background that the bacteria are stuck to, is totally clear. It did not uptake any color. So the bacteria take up the color themselves, positive from the inside, and the background stays negatively stained. There's no color there, so a positive type stain. This, on the other hand, is a negative type stain, whereas the bacteria themselves did not take up any of the stain or any of the dye, and the background did. And the bacteria stand out against the background, whereas these guys over here, the bacteria are, the background's clear, the background's colored, and the bacteria are clear. So this is the opposite. So positive type stain on this side and a negative type stain over here. Positive type stains are very commonly used in research labs and schools and things. Negative stains, not so much. So one of the most common types of stains that's used in a microbiology lab is something called a simple stain. So simple stains are really easy to understand. What this is it's a type of positive stain where you heat fix the bacteria to a slide and you add 1 or 2 drops of usually some sort of substance that uses a charge to stick to the bacteria. So usually a positive charged dye, commonly methylene blue, as this is methylene blue down here that's been used. It's blue, hence the name. And that will has a positive charge, and it sticks to negatively charged things like DNA inside of the cells. So that's most of the time what a simple stain does. It sticks to charge. So a simple stain is useful, but it is, however, limited in the information that it can give you. What it does is it shows you the size of the bacteria. Now you're going to have to look at quite a bit of bacteria underneath a microscope to be able to judge if a bacteria is large or not. If it's a big cell or a small cell, you have to look at quite a few of them to be able to tell that. Whereas if you look once you look at quite a few of them, you'll just be able to judge right underneath the microscope the very first time. Um, so it gives you size. It also gives you shape. There are a couple of shapes of bacteria we'll talk about later. So you'll be able to look at the microscope and go, oh, this is a circle, this is a rod, this is a spiral shape. So it gives you size, it gives you shape, and it's also going to give you something called arrangement. So this is going to be how the cells are stuck together. Are they stuck together by them in a group, a blob, a straight line? Are they not stuck together? Are they stuck together in groups of fours, groups of twos? Are they by themselves? And things like that. So that's called the arrangement. So a simple stain gives you something useful information, but it only gives you size, shape, and arrangement. So those three things you get from a simple stain. It's limited on the information that it gives you. So differential stains, on the other hand, a gram stain happens to be one of these, will give you size, shape, and arrangement. It is a stain. Every single type of stain will give you that size, shape, and arrangement. And this is no different. So it will give you size, shape, and arrangement, but what a differential stain will also give you is an extra piece of information that will allow you to tell something extra about the bacteria that you've just stained. So it will tell you, oh, this is a different type of bacteria than that one, different species, or oh, this one stains different. Oh, this one doesn't uptake this same type of dye, so you might want to look at this one. So you guys can see down here in the corner, we've got pink and purple. This is a gram stain. So the purple bacteria uptake a different type of dye than the pink ones. So we can differentiate apart that this is, oh, it's different. Something's different here. Whereas if you used a simple stain, you would simply get the size, shape, and arrangement, and everything would come out blue. You would not be able to go, oh, there's two different species here. Everything would just look the same. It's all blue. It's all the same shape, all the same size. Whereas when you use a differential type stain, like a gram stain, you get size, shape, and arrangement. Oh, they're all rods. Oh, they're all about the same size. They're all the same shape. Oh, wait a minute. But they've got that extra little bit of information that allows us to tell apart that these are two different species, whereas we would not have that from the information earlier. So size, shape, arrangement, and an extra piece of information, which is usually going to be gram reaction or acid reaction or something like that, whereas the simple stain only gives us size, shape, and arrangement. So gram stain is the most commonly used type of differential stain. This is so common that hospitals actually have machines that do this. You, as a lab technician, would just put the sample on the slide and then hit a button, and it'll do it for you. So gram stains are broken up into two different outcomes. You have gram positive, which is purple, always purple. The bacteria, if you do this technique properly, that are gram positive will come out purple. Bacteria that are gram negative, as you see over here, will come out pink. Now you can have gram positive rods, gram positive coccuses, gram positive spirals, every other shape of bacteria, and same for gram negative. Shape does not matter, size does not matter for which one of these. You can have the same size shape in both. So what does this mean? What does this matter? I'll explain in a second, but what does this really mean in the short, sweet terms? Antibiotics that work for these guys, gram positives, tend to not work for gram negative bacteria. And antibiotics that work for gram negatives tend to not work for gram positives. So if your patient comes in and they're dying very quickly, you need to figure out what they've got. You can go, oh, they have a gram positive infection, hang an antibiotic up, and it'll probably work well enough to buy you some time to figure out what the exact infection is caused by. Whereas if you just go, oh, they have an infection, and just hang up an antibiotic, you got a 50-50 guess of it not working. So quick and easy test to break apart which type of antibiotic to use. This takes about 3 minutes, this type of test, if you do it correctly and if you're good at it. So you can figure out your results very quickly, which type of antibiotic to start with. Like I said, the antibiotics don't work on the other classes. So very, very, very important thing to do. This is the very first step for diagnosing any type of infection, any type of bacterial infection. You have to know what its gram reaction is. So I'll talk about this actual process of gram staining in lab. This is kind of a short little quick thing of how it works. I'll talk about it in more detail. What it does is it works off of the structure of the cell wall. So gram-positive organisms have a such cell wall that has a totally different structure, as you guys can see over here, than gram-negative. So this is what this looks at. So the stains react differently with each type of cell wall. So the different type of cell walls produce either purple for gram-positive or pink outcome for gram-negative, depending on the type of stain that they uptake. So there's another type of stain that's used a lot in microbiology labs called an acid-fast stain. And this type of stain is used a lot for organisms that have a really, really, really difficult time staining any other way. So these guys, these particular species, there's a couple of them. Mycobacterium is a genus that tends to be acid fast. This is the organisms that cause Mycobacterium tuberculosis, leprae, leprosy. Um, so um, Tuberculosis being the most common one. As you guys can see down here, it's as it grows on a petri dish. This is actually it right here. It looks kind of like candle wax, like somebody spilled a candle. So if you've ever put water on a candle, you know that the water just beads up off the wax. It doesn't penetrate it. So unfortunately, the same thing happens here. And if you try to stain these organisms with crystal violet or gram stain techniques or anything like that, if you just try to pour liquid stain on them, it doesn't penetrate down in their cells. So you have to use a special type of stain that bonds to their that can break through that waxy coating. And that particular type of stain is called adheres to something inside of their cell walls called mycolic acid. So mycolic acid is that waxy coating here. It prevents the stains from binding. So you have to find something that bonds to it. It tends to be a dye called fusion. So acid, since the mycolic acid, you have to find something that bonds fast, holds fast to that acid. So that's where this term comes from. So what this is, you guys can see down here, tuberculosis. If you do a gram stain technique properly, the cells from the tissue of the human, this is lung cells, come out green, and the bacteria come out bright red from the background. So you can see the tuberculosis stand out really, really, really obviously from the cells of the human lung tissue themselves behind it. So this type of bacteria, these species, would not be able to be gram stained. If you tried to gram stain this cell, this human tissue over here, they just would not uptake the dye. You wouldn't be able to see them. So you have to use different types of techniques to see, to look at the organisms when you suspect a tuberculosis infection. So this is a structural type stain over here. And this is looking at the presence or absence of a capsule in bacteria. So what a capsule is it's kind of a little protective coating that bacteria can produce for themselves. So you guys can see here, this is the bacteria itself, the little tic-tac in the middle here. And this capsule is essentially just a little blow-up balloon, a little zorb, if you like, of sugars and proteins and junk that the bacteria has produced that grows all around it. So you wouldn't know that that was there. You can't see them underneath a microscope. They're clear. So what you have to do is you have to stain the background. So this thing does not absorb stain. This capsule doesn't soak up colors. It's really dense. So what you see is the bacteria takes up some color. This thing doesn't. So you see the background that's stained, the bacteria that sucked up some color, and then a big empty colorless blob there where the capsule is located. So capsules, what they do is they do a lot of stuff. It helps the bacteria stick to the surface of cells or the host organism that it wants to cause disease in. It can cause the immune cells to not be able to destroy these type of cells. They can't eat the capsule, which is kind of cool. So the bacteria is able to survive the process of phagocytosis of being eaten. So lots of gram-negative rod species tend to produce these. Not every species of bacteria can produce them, and just because you're a gram-negative rod doesn't mean you can produce them either. So if you want to look for a capsule, you would not be able to see these underneath a regular gram stain, since the gram stain just stains the colors of these and not the background. So you have to use special techniques to be able to see them. Endospores. If you recall from lecture 1, endospores were the thing that messed up Louis Pasteur's experiment trying to disprove spontaneous generation. Microbial generation, I should say. Endospores are pretty much little indestructible, little bulletproof vests that are found in bacteria. They can't be destroyed by boiling. You can only destroy them by fire. They don't just get destroyed by freezing. They pretty much can survive everything. They have a very thick cell wall that surrounds them, very, very, very thick coating that doesn't allow stain to penetrate. So what you have to do is you have to heat them up with boiling water, which causes their cell membranes, the cell walls, that very thick endospore coating, to kind of loosen up a little bit, to kind of relax, which forces stain inside, this green stain called methylene blue. So these bacteria here are red. The endospores are green. So you can see here, this is all these little green endospores have been released from the bacteria. I'm trying to see if I can find you guys one. There's one right there. You can kind of see the little green speck still inside of the bacteria. It hasn't been released yet. So all the green ones have been released. There's a couple down here that haven't been released yet either. One right there. All the green ones that have been released from the bacteria into the environment. So you have to use heat and a very specialized form of stain that's able to penetrate through that very thick cell coating around those endospores. Flagella stains, if a bacteria has a flagella, you might want to see that. You see how many they've got, how many there are. So you have to use a specialized stain that can stick to the types of proteins that are found on flagellas. So anyway, on to the 6 I's of microbiology. We'll talk about these fairly briefly. They're pretty easy, and we'll walk through them. So the very first step of microbiology lab, of getting a new patient sample and figuring out what it is to isolate away the sample from your patient that actually is the organism that's causing the problem. So when you get a sample from your patient, you're not just going to get, oh, here's just the swab with just streptococcus growing on it that's causing the strep throat. What you're going to get is a sample with a swab with strep throat on it, yeah, of course, but every other bacteria that grows in their throat, of which there are quite a few of them. So your job as the microbiologist is to go, which one of these is causing the problem, and figure out how to get away just that one species from all the rest of them. So this process is called isolation. A very common technique is a process called streak for isolation, where you take it's essentially like imagine having a bunch of marbles in a cup, and you throw the marbles across the floor, and all the marbles roll across the floor, and they all spread out as they go. So that's what you do. You take your swab and you spread it out all over a petri dish. So all the bacteria that would be grouped together from your patient on the swab are now spread across that entire petri dish. So they'll grow spread out, separated apart from one another, and then you can get the individual cells. So as you can see down here, same thing, bunch of blobby cells all put together. You spread them out all over the petri dish, and then they grow separated apart from one another. So you can scoop this little guy up and test him, scoop this little guy up and test him, scoop this little guy up and test him. Where if you have a big blob of cells all growing on top of one another, you're going to get mixed, conflicting results, because these guys might be gram-negative, these guys might be gram-positive, and then who knows what all the other results are going to give you. So the very first thing is to isolate away just one species of bacteria that you can test to give you consistent results for just one species. There's quite a few ways to do this. The most common one is something called a streak plate, or an isolation technique, streak for isolation technique, where you use a very specialized process of streaking bacteria around and on a plate in a certain angle, certain way. There's lots of different ways to do this. And what you do is, you can see over here, you got a bunch of bacteria all condensed all together. You could not get in there and figure out what colors, what species there were if you needed to. Spread them out this way, and they're starting to get a little spread out. You can start to see some red in there. You can start to see some yellow. And you just keep going, keep going, keep going, and then they get more and more spread out, whereas, oh, there's a little white kind of clearish colony in there. You can even see these over here. You got red, white, and red, yellow, and kind of that whitish clear. You can see these over here, that they were there. So what you can do is you can go in here and scoop this little yellow guy up, all one species, scoop this little red guy up, all one species, scoop this little guy up, all one species. Test them for their independent results, get reliable, repetitive results, and figure out what's going on. Whereas over here, if you just take this sample and try to test that, you got all 3 of these species mixed together. Who knows what you're going to get? You have something called a pour plate technique. You essentially just take a big liquid plate, mix them together, put the sample in, mix it all together, spread it all out evenly, and then pour it out on a petri dish and let it dry. So the bacteria are mixed in, they spread out evenly, and then they get spread out on the plate when you dry. You can also just mix them around evenly on the plate, spread them around on the surface, and they'll spread out that way. It's the concept, once again, of throwing the marbles. The next I is inspection. You got to look at them and see what you're looking at and make sense of what you're looking at. So one of the very first things that you need to check for is, once you start growing things in a microbiology lab, is contamination. Is what you're looking for really there? Or do you have some extra friends there that you don't want? So you can look over here and you can see, oh, there's a lot of yellow cells. Those are awesome. But we have some extra little friends that are mixed around in there that we don't want. These little kind of clear white guys in there. These little kind of opaque, kind of milky colors. We don't want those. Those are contaminations. This is bad. If you try to use this culture, this plate for diagnosis, you're going to have two species mixed in together. So you're not going to get reliable verifiable results. You're going to get conflicting results from those two species. Now over here you can see again, you have contamination growing. You can't use this plate. It's messed up. It's useless. Whereas over here, oh, all one color, all looks the same, all one color, all looks the same, all one color, all looks the same. These are the same. It's mixed cultures over here, pure cultures over here. So you need to inspect it to make sure that what you see is what you really want to see. Do all the little bacteria look the same? Are they the same shape, the same color, the same size? Are all the colonies the same? Or are they different, whereas they're over here? These guys are nice and round and red. This one's kind of edges are kind of a cloudy, kind of chunky. It's white, it's fluffy looking. Over here, are these all nice and yellow and round? No, they're all round, but these aren't yellow. So these are obviously two different species, These are all good. The next one is identification. So once you've isolated and inspected your samples, you want to identify what you're looking at. And there are tons of different ways to do this. You can stain them underneath a microscope to see what they are and get a little first step there. You can send them off to have their DNA sequenced and figure out what it is. You can do lots of different tests on agars and things like that. You can run different immunological tests looking for antibodies and things like that. There are tons and tons and tons of different ways to identify bacteria, and we'll talk about a couple of them later on. So one of the most common tools and ways to identify bacteria in a lab is using media. So media, what it is it's essentially food for bacteria. So you put bacteria on a petri dish or something, and they got to have something to eat. So whatever that something to eat is media, is what that is what it's called. Media comes in lots of different flavors, forms, different properties and things that are used for different applications, but essentially it's just bacteria food. And it comes in three different, you classify it based on three different ways. Its physical state, how it's put together, and what it's used for. And this makes sense if you think about it. So physical state are really easy. If it's liquid, broth, or solid, or semi-solid, excuse me. So liquid, it makes perfect sense. If it's essentially just chicken broth or beef broth or something like that. It's a liquid media. It never turns solid. No matter how long it sits there, it will always be liquid. So this allows you to keep bacteria that can swim around, or you can allow bacteria to tell different how they like oxygen, which we'll talk about later. You can see here, there's not a lot of bacteria towards the top. These guys don't like oxygen. These guys do like oxygen. They're all gathered at the top. These guys like just a little bit of oxygen. These guys can move around. These guys can't, blah, blah, blah. Whereas over here, you'll see a semi-solid media, which is kind of like Jello. It's sort of solidy, sort of liquidy. These can allow bacteria to move around. You can see and hear these guys can't move. They're exactly where you stuck them in a little streak. These guys could swim around and swim through the agar. This allows you to see if bacteria can move or not, which is a big important thing. If you get an infection, are the bacteria stuck where they're at, or do they have the ability to swim from, say, your eyeball to your brain and cause problems that way? You kind of need to know that information. So this type of agar allows you to see that. And then the most common type of agar that is used in labs are petri dishes, solid agars, where these are more like Jello with lots of gelatin in them. They're solid. You can physically touch these and shake them around. They're going to stay where you put them. They're not going to fall out of the plates and flop around. So this allows you to put bacteria on the surface, and it's going to be a fairly stable surface for bacteria to grow on. That allows you to work with them instead of having to risk spilling them over here with our liquids or them sloshing kind of around with the semi-solid. So each one of these has different purposes. They're all three used for different things, and no one of them is better than others because they literally all have different purposes that you use them for. So once again, it's comparing apples to oranges. They're all used for different things and all have different uses. So essentially, what it is they're all made up of something called agar. So media is essentially Jello. If you guys know anything about Jello, it has gelatin, which is the solidifying agent that makes everything nice and squishy and solid inside. And then you add your flavoring agents and water and things like that, sugar and whatnot. Agar is essentially the gelatin for microbiology. They used to use gelatin from beef, but unfortunately, animals or bacteria can eat gelatin; they can digest it. So when you put bacteria on a plate that's made of gelatin, they ate their home, and then eventually, what would happen is they ate their petri dish, and then they would starve to death, and that's not very good. So we had to come up with something better. So agar is used nowadays. Agar is a derivative from seaweed. The red algae, Rhodophyta, is the phylum of seaweed that produces that. It's also the same thing that they use to wrap seaweed in, or sushi, I should say, nori. It's actually red when they harvest it. It dries out and turns green. So if you guys see down here, this is used a lot in cooking. It's called agar. They use it a lot to make clear gelatins and things like that. It has no taste. It's also vegan friendly. So it's kind of the vegan gelatin, but they use it in microbiology. It solidifies up, and instead of adding, say, lemon flavoring or that kind of stuff, you add in different types of salts, different types of proteins and things like that, different types of chemicals, sugars, and whatnot that different types of bacteria can eat. And the different types of combinations of sugars or salts and things will kill certain species, allow certain species only to grow and things like that. You can add in antibiotics, pretty much endless combinations of stuff that you can add in to the Jello to make different types of petri dishes that all do different things. One of the most commonly used types of agar in a microbiology lab is nutrient broth or nutrient agar. So nutrient agar, nutrient broth, essentially is just made by boiling a bunch of beef bones, a bunch of old meat and things like that, meat scraps, or a bunch of soybeans and things like that, wheat and things like that, and you boil it for a really, really, really long time to bring out all the proteins that are found inside of it. So this is essentially just like chicken broth or beef broth and things like that. So the bacteria, you will introduce them to this type of media. This is a general purpose media, which means that pretty much every type of bacteria can grow on it. It's not a specialized use. It's not meant to only grow Streptococcus or maybe one or two types of species, or not meant to grow just gram-positive species or just gram-negatives. But this type of media can grow pretty much everything you put on it. General purpose. So you can see here the liquid form. The broth form, it's just essentially chicken broth or beef broth or things like that. You add the agar to it, and it becomes solidified like Jello, and you can pour it in a petri dish. So once again, general purpose agar used a ton in microbiology labs just to grow things, teach people how to use plates, practice, and just keep cultures alive. So the other way that you define agar is what's inside of it. So is it going to be something that you can look at on the back of the agar, the back of the bottle, and go, I can look at every single ingredient inside of this thing and know the exact precise formula of it? So there's a very common type of media that we'll talk about in the lab called mannitol salt agar. Mannitol is a type of sugar. Salt is sodium chloride. And you can flip around on the back of that and it will tell you there is 12.5% mannitol, 7.5% salt, 8.6%, blah, 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 so on and so forth. And it will tell you every single ingredient and how much of that ingredient is in that agar. That is something called a chemically defined agar. You know exactly what and how much of it is in every bit of that agar. That's important to know if you want to have very consistent, very reliable results that will always work in a microbiology lab to grow exactly what you want every single time. And then there are other types of agars that are called complex. And these are things, something like a tryptic soy agar. Tryptic soy agar is made from soybeans. Nutrient broth is kind of the same way. It's made from beef broth. Oh, I don't know what makes up a soybean. Nobody knows what makes up a soybean because every single soybean is different. Every single soybean has a slightly different content of nutrients inside of it than every other soybean out there. So you can't say for 100% fact that this agar contains this amount of stuff. You just can't do it. So those types of augars are called complex. You might know most of what's inside of it, but the soy part, the soy proteins or the beef proteins, you just don't know. So that's what's termed complex. You have general purpose versus enriched media. So general purpose media is things like nutrient broth, tryptic soy agar, blood agar, where pretty much, not blood agar, where pretty much everything can grow on it. You put a sample of your skin bacteria on nutrient agar, pretty much every single one of those species is going to grow. It's a complex media, tryptic soy, nutrient broth, contains beef broth, beef peptone agar, BHI, brain heart infusion, all that kind of stuff. It just grows pretty much every single bacteria that'll grow on it. If it's gram positive, gram negative, if it's a coccus, if it likes to eat sugars, if it likes to eat proteins, it doesn't matter. It'll pretty much make them happy and they will grow. Whereas an enriched media, on the other hand, has some sort of specialized complex ingredient inside of it, synthetic ingredient, that usually only allows one or two species of bacteria to grow. Some bacteria are considered something called a fastidious organism. You guys can see this term down here, which means that they require special things to grow. They require salt in their diet, or they require iron in their diet, or they require a certain amount of protein that maybe not is going to be able to be found in a nutrient agar and things like that. So if you want to grow these types of bacteria, you have to give them specialized media that gives them the stuff that they have to have. So fastidious microbes require certain types of nutrients that general purpose medias don't usually have. So if you want to grow a fastidious organism, you have to give it those specialized growth factors in the form of an enriched media. So same thing here, chemically defined versus complex. So selective and differential. So this is kind of a step back a little bit. So any type of agar can be selective and differential that's not general purpose for the most part. There are a couple of general purpose that are differential, but not always. So what this concept is, selective medias will allow, general purpose medias will allow pretty much every single bacteria species to grow. They're not going to be able to tell apart different species. They're going to allow everything on there to grow. Pretty much everything that touches it will grow. Whereas a selective media, if you take a sample from your patient that's mixed together and you add it onto a general purpose media, you guys can see over here, everything in that sample is going to grow. A selective media, on the other hand, is going to have some sort of ingredient inside of it that's going to kill most of the stuff in here that you're not concerned with. Let's say you think as a doctor they have a staph infection. Staph really likes salt. Doesn't love salt, but it can tolerate salt. Most of the other bacteria that are going to be found on humans or in humans can't tolerate salt so well. So what you do is you take a sample from your patient and you spread it on an agar that contains lots and lots and lots of salt. What's going to happen is the bacteria that can't tolerate the salt are going to die. And if there's staph present, they're going to be able to survive and grow. And you're going to go, oh, there's not supposed to be staph there, but there is. I can see them because they grew. All the rest of the stuff that's supposed to be there died. The only thing that grew is something that's not supposed to be there. Now I know what they have. So selective media uses some sort of property about the bacteria to allow it to grow while killing off pretty much everything else we're not interested in. Differential is the opposite of that. Everything can grow for the most part, but it allows you to tell them apart based on if they can eat a sugar or a protein found inside of this agar. Maybe there's 4 or 5 different types of dyes, and this guy uptakes the blue one, this species uptakes the green one, and that's how you can tell them apart. So selective agar will kill everything but a couple of species, and that's what you're going for. You want to just see the species that grow. Those are the ones you're interested in. Differential media doesn't kill everything, but what does grow, you can tell them apart. So just if you are a selective media, you are differential. You have automatically selected against some sort of species of bacteria because not everything's growing. But if you're differential, you're not always selective because you might allow everything to grow. You can just tell them apart, but you didn't kill anything. Everything grew, but you can still tell them apart. You didn't kill anything. Just because you're differential doesn't mean you're selective. But if you are selective, you are automatically differential. So here's a good example of that. So this is mannitol salt agar. It is selective and differential. So this type of agar is selective against gram-negative organisms. So it kills gram-negative organisms. It has a lot of salt inside of it. They don't like salt. They dry out in the presence of high concentrations of salt. Whereas most gram-positive organisms can survive in high presence of salt, especially things like staph that grow on your skin. So what happens is you introduce a sample of bacteria from your patient, and any gram-negative organism is going to be killed. This is Micrococcus luteus. It's a gram-positive organism. It just doesn't like the salt so much. But this is what gram-negatives would look like once you introduce it to this agar. Everything died. There's no change. Nothing grew. Everything was killed by the salt. So that's the selective part. It killed everything except for stuff that likes salt. They're just gone. If you can grow, you like the salt. That's the selective part. You're okay with the salt. It doesn't bother you. So over here we have Staphylococcus aureus. You can obviously see since it grew, it doesn't mind the salt. The salt doesn't bother it. But this guy is also Staphylococcus over here, Staphylococcus epidermidis. He liked the salt, doesn't bother him, so he grew. But they are yellow and pink, different colors. This is the differential aspect. This guy can eat mannitol. This guy can't. Once this guy can eat mannitol, he produces an acid, which makes this agar yellow. This guy can't eat mannitol. He produces a base. He eats the protein, produces a base, which makes the agar kind of pink. This is the differential aspect. The selective aspect is they can grow. This guy can't. The differential aspect is now we can tell apart who grew based on the color reactions of the agar. You can diagnose the species. Same thing over here. The different color combinations, who can, who can't grow, things like that, will tell you what the species is. So one of the most important parts of getting a microbiology sample is getting something called a pure culture. So when you get a sample from your patient, I mentioned this already, you're not just going to get one single species from them. That's very, very, very rare in nature. You're not just going to find one species of bacteria growing by itself. As you guys know, you usually don't just find one species of animal there. You find multiple species of organisms living together, and that's pretty common. So you're going to take a sample from your patient, and you're going to get samples of bacteria that live all over their skin, all over the inside of their nose, different species, species from the environment, and then you'll also get the sample of bacteria that's causing them problems. Now, your job as a microbiologist is to get away the bacteria that's causing them problems from the ones that aren't, because you want to figure out which one's causing problems and which one isn't, because they all look the same. It's kind of hard to do that. So your job is to figure out how to get something called a pure culture. Now, we talked about that earlier with the concept of isolation. You spread them out, separate them apart, go for the colonies that are separated and things like that. But what a pure culture is it's one single species of bacteria growing all by itself on a petri dish. So these are really useful for diagnosis. This is what you want. This is a pure culture plate. This is a mixed culture plate. So you can tell just by looking at it. All the bacteria that are growing on this thing, by the way, this is a colony. A little circle of bacteria, it's a little group of bacteria that are all genetically identical. That's a colony. So all the colonies on this plate are genetically identical to one another. You can see that they're all the same shape, they're all the same size, they're all the same color. Pretty good chance that this is a pure culture plate. You could take a sample from here, a sample from here, a sample from here, anywhere on this plate, and run tests on it, and you will get the exact same results every single time, because all of these bacteria are the exact same. This is what you want. This is useful for diagnosis. This is what you have to get reliable results for your patient to tell the doctor what they have, or if you are the doctor, to diagnose what your patient has. Whereas over here, this is a mixed culture plate. If you take a sample from here, a sample from here, sample from here, sample from there, there are multiple different colonies on this plate. You can tell they're different shaped, different sizes, different colors. They look different. The edges look different. All of these guys look totally different. Anywhere you sample on here, you're going to get different results. Different species are going to give you different test results. This is not useful for your patient. You might kill them trying to diagnose them over here, whereas over here you have one species. Fairly easy to get reliable test results on that one. This is the whole point of a microbiology lab, is to get that pure culture. So once you get that pure culture, you're usually going to start out with something called a mixed culture plate. So you usually are going to start off like this with your patient. Your job is to get away, pick off these little isolated colonies, and do something called subculturing. So you'll start off with a mixed culture plate, and then you'll start with the isolated colonies. You'll scoop up one of those and subculture him into a new pure culture plate all by himself. So what you'll do is you'll take all the different colonies. So let's go back over here, and you'll scoop this guy up by himself and start his own pure culture plate. Scoop this guy up, start his own pure culture plate. Start this guy, scoop this guy. Every single isolated colony that's different, you'll start them up their own pure culture plate. And that allows you to get pure cultures of every single species that was found on your patient. And then you can diagnose which one was causing them problems. So that's the concept of subculturing, figuring out who's inside of there and growing up those individual little samples. So the end of these lectures are going to be really, really, really easy. And we're just going to talk about some common infections that you guys might find in a microbiology lab or in a healthcare setting and things like that. So how this is set up, as you guys will see up here, we have the disease itself, cholera, and then the organism that causes it, Vibrio cholera. This is the bacteria that causes cholera. So this is the bacteria itself underneath the microscope. It's a Vibrio shape, a little kind of curved rod, and it's got one single flagella on the end. So cholera, you guys will see a couple of pictures, maybe one over here and one over here, usually that are relevant to cholera, some things about it. So cholera is a gram-negative, comma-shaped, waterborne pathogen. It's spread by water. So usually what happens is people will come down to crowded banks of rivers and streams and things like that, and they take baths, but they also defecate in the water, or maybe they toss their sewage in the water and things like that. So the bacteria are passed by feces. So someone that's infected with cholera passes cholera bacteria in their feces. They toss their water in that feces in the water. The bacteria replicates in the water. So someone else comes along, takes a big old sip of that water to drink, splashes around, accidentally gets some in their mouth and their nose and stuff. And then cholera is able to spread that infection to the new person. And that's how it spreads around. It used to be extremely common, killed tons and tons and tons of people all across the world. Nowadays, it's kind of relegated to a lot of developing countries. Every time there's a disaster, after Hurricane Katrina, there were countless cases of cholera in Louisiana. So it always occurs after massive natural disasters where there's a lot of sewage leaks and things like that. About 2 to 5 days after you drink some nasty water, get some feces contamination in your mouth, you will start to develop some stomach cramps, muscle cramps, things like that. Large amounts of non-bloody, watery diarrhea, vomiting, things like that. The diagnosis for this is a stool test. They will take a feces sample and look for salmonella inside of it. There's really not a lot of treatment they can do for this unless you're really, really, really severely sick. Most of the time what they're going to do is to just give you maybe some IV fluid replacements if you need it, or drink some Gatorade. Keep your fluids up. You're going to be losing a lot of liquid, a lot of hydration through dehydration and vomiting, so you just have to keep yourself hydrated. That's how most people die from cholera. They're just not able to keep any liquids down, so they're dehydrated and they die. If it gets really bad, they'll give you some IV antibiotics to kill off the bacteria, but it doesn't usually get that far. About 28,000 deaths on average per year across the world, and about 3 to 5 million cases per year end up in the hospital with cholera, according to the World Health Organization. So leptospirosis, anybody that's a veterinarian will encounter this at one point in time, very commonly found in animals, usually called lepto. It's a corkscrew-shaped bacteria. Very interesting little organism. It's spread by the urine of rodents. So if you come in contact with rats or things like that, a lot of dogs and cats will get this, coming in contact with rats and things and squirrels and whatnot out in the woods. Hunters will get it from time to time through the same process. Well, anyway, you come in contact with the bacteria in the urine. When the urine's moist, it's obviously a little more infectious. As it dries out, it becomes less infectious as the bacteria start to die. Well, anyway, you come in contact with the infected urine, it gets inside of your body. What will happen is these guys will burrow, they're corkscrews, they work like little drill bits, as they'll burrow inside of your muscles, inside of your organs, and things like that. They'll eventually lead to bleeding inside of the lungs, kidney failure, things like that. They can cause brain problems if they get inside the meninges, which is a big deal. They can get up that way, spread around by blood. Fairly easy to treat if you catch it early. It's fairly common in other countries. Haemophilus influenza, I think this is our little last one. Hib B is the vaccine that most children get for this one. This was at one point in time thought to be the causative agent of the of the flu before the flu virus was identified, hence the name. The symptoms are essentially the same for the flu. It's a little different. It does cause swelling of the arms and things like that, which is one of the different identifiers from the flu of this particular disease. But all the rest of the symptoms are essentially the same. Is resistant to most penicillin families, so you have to use a different type of antibiotic on this one. It's not the most uncommon thing on the planet, but it is slightly common in the United States. We do vaccinate it for it, though, and it is, oddly enough, the very first organism to have its entire genome sequenced, which is kind of cool. The very first free-living organism to have its entire genome sequenced, which is kind of neat. Oh, I also forgot to mention, if you see the little vaccine symbol down here, there is a vaccine for this particular disease. If it's not there, there is not one. So that's the last little bit of this one, guys. That's all I've got for this lecture. If you have any questions, feel free to email me about it. Other than that, y'all take it easy.